to get started. So with that said, thanks all for coming. This is our third in this new format. Maybe not the most smoothly running one yet, but we'll get there. So I'd like to thank Ross for taking the invite to come and give this talk, which he was scheduled to give in person at the Douglas. So Ross is a third year PhD student, sorry, in Bryce Missions Lab at the MNI. And I'm on his PhD committee, and kind of this invitation was born out of some presentations I've seen him give through that committee, and I thought his work is really relevant to a lot of the challenges that a lot of us are facing right now in terms of making choices about, you know, supervised machine learning or unsupervised machine learning methods, really, and multivariate methods and design implementations and jitter that you might make when you make a design choice, ripple effects that might have down the chain of MATLAB or R or Python commands. In Ross's case, definitely Python commands that you might you know, that you might choose to make. So before that, he was a RA for 3 years at Cornell, right? And did and has, as he said previously, learned to code out of necessity. I would argue he's very good at it. So with that said, I'll let Ross take it away. And thanks again for taking the invite to come and give us a talk.
Yeah. Well, thanks for having me. Thank you everyone for showing up. Hopefully you are all comfy at home with coffee, etc. Sorry about the dramatic change of Zoom room. So yeah, I am really excited to talk to you about some of the work that I've been doing over the past year or so. Looking at how as Millar kind of described, the impact of certain methodological choices that we make can really dramatically change the analyses or results of the research that we're doing. And so Millar mentioned some things with machine learning and some unsupervised, you know, that sort of stuff. I'm actually not going to be talking as much about that today. Those are projects that are kind of in the works, but hopefully I'll still have some interesting things to say. So to get started, I just want us to kind of all think about what a typical research project for a lot of us might look like. And so the way that starts off is with us developing a research question hypothesis, whatever that may be. We get our data somehow, I'm gonna breeze over that a little bit. We pre-process our data, we run whatever analyses we might have, and we generate some statistical assessments, and then very hopefully we publish our results. And so at each of these points, we're making a number of different decisions, but throughout the talk today, I really want to highlight these middle three steps: pre-processing our data, running our analyses, and generating statistical assessments, and the choices that we make in doing those things. Obviously, there's the other steps all have different choices. Where do you want to publish your results? I would recommend a preprint server followed by an open access journal, but that's my bias. How do you get your data? There's obviously a number of different ways to do that. And then there are, you know, an infinite number of different research questions and hypotheses we can come up with. But I think in terms of the methods that we're using, these middle three steps are really critical and influence a lot of the work that I'm doing. So I'm not the first person to think about the idea of methodological variability or the choices that we make during the research process. This has been, you know, covered at various angles throughout history of research. More recently, there's been some great work looking at, for example, different choices that we make during pre-processing and data preparation. Some work from Lauren Parks out of Alex Fornito's group a couple of years ago looked at different motion correction techniques in fMRI pre-processing and found that all of the different techniques that are used by the field will influence the reliability of the signal that we get out of our fMRI data quite dramatically. Just a couple, I think last week, Stuart Oldholm, also out of Alex Fornito's group, published a paper looking at diffusion MRI processing. And what they found is that running a number of different pipelines, the results that we get, for example, in this case, nodal strength, so how many structural connections does a given brain region have to the rest of the brain, differs dramatically as a function of the processing pipeline that you're running. In data analysis, this has been looked at. Alex Bowring, Camille Lemay, and Tom Nichols, last year, they presented this work actually at OHBM, and it was published in Human Brain Mapping. They looked at running the same analysis using 3 different software packages. So in this case, AFNI, FSL, and SPM, which are 3 of the kind of most widely used, arguably functional processing software packages in the field. And they found that doing the exact same thing in those 3 different packages yields dramatically different results that are kind of, you can just see. There are consistencies, but there's also significant enough variability that the interpretations you make would be quite different. And then with respect to statistical assessments, last year, about over 70 different research groups got together to do this NARPS study, the neuroimaging analysis, replication, and prediction study. And what they found is that everyone was supposed to do the same thing. They proposed the analyses that you were supposed to run, and then everyone was supposed to generate statistical maps for different kind of canonical task fMRI contrasts. And they found that the reported activations for these contrast maps varied hugely across the 70 different research groups that participated. So what we see here is just a function of the percentage of research groups that reported a significant activation in a given voxel. So 1 is all of the research groups. And you can see that there are very, very few regions for some of these contrasts that approach that. So throughout all of these stages, you know, these results and research groups have highlighted that the choices we're making are leading to very different things. So that idea of methodological variability, it's everywhere. So if you will, imagine kind of a research pipeline. You start off with, let's say, some brain data. You've chosen a pre-processing technique and you have your functional time series for, let's say, a group of Parkinson's patients. You have a brain atlas. You've decided, I want to divide up the brain into these distinct anatomical regions, and I'm going to use those to construct some brain correlations. And, you know, I can maybe look at the differences between these patients and healthy controls, or I can start to get, you know, add in some other data sets. Let's say I want to add in some other brain data, like gene expression data from the Allen Human Brain Atlas. I can do some more brain correlations, and then I want to make statistical inferences. So the top row here is kind of what I just covered in those things that people have done. But significantly less attention in my mind has been paid to this bottom row, the other data sets that we're bringing into our research analyses, the sorts of statistics we're running with this multimodal brain data, and the inferences that we're making from it. And so my work is going to focus on highlighting some variability in the methods that we're using for these sorts of analyses and how that can have some pretty dramatic effects. So I'm going to start off talking about pre-processing and data analysis, and I'm going to specifically highlight transcriptomic processing pipelines. And so when I refer to transcriptomic processing pipelines, there's a lot of different transcriptomic neuroimaging data sets that are kind of coming to the fore, but the kind of tried and true in my mind is the Allen Human Brain Atlas. It was released now a decade ago. I think the first release was in 2010. And if you haven't heard of it, they collected 6 brains from postmortem donors. And from those brains, they took 3,700 distinct tissue samples from all over the brain. And they used microarray probes to index the expression of about 20,000 unique genes all over the brain. So it's an incredibly spatially comprehensive map of the genetic architecture of the human brain. And they've made this data all openly available, easy to download, accessible online. They have a number of tools supporting access of this data, exploring it. I would highly recommend that you look into the website, humanbrainmap.org, if you've never been exposed to it. But it is being increasingly used in research. It's been out for 10 years now, so it's seen a lot of use already. In 2015, they looked at the impact of, you know, functional connectivity within resting state networks, the impact of gene expression on these functional connectivity values within different canonical resting state networks. A year later, people looked at how gene expression influences different topological properties of brain networks. They've looked at how gene expression values across the brain differ and how that mirrors changes that we see during adolescent development, and then what gene expression in different regions can tell us about the functional specialization of different brain regions. So it's kind of spanned the gamut, the use of this Allen Human Brain Atlas in research. This is a figure taken from that last paper by Kevin Anderson down at Yale. He's a graduate student. I believe he just finished his time there. And they looked at the relationship between gene co-expression, that is how similar the gene expression is between different brain regions, and functional connectivity taken from BOLD. And they see that there's a lot of concordance between those maps, although there are some notable differences that methods matter. The methods that we're using, the choices that we make in our research are important, but not always in a predictable manner, as we saw. And so everyone's question is always, what do we do? Besides the immediately obvious, which would just be to panic and then not do anything. And so we have some options. The first option in my mind is this idea of pre-registration. If you've never heard of pre-registration, this kind of overview by Brian Nosek and colleagues published a couple of years ago is great. Also, you can go to the OSF website, learn all about it. But briefly, pre-registration is this idea that you're going to do a single analytic stream, and you are going to tell people ahead of time what your analytic process is going to be. You come up with your idea, you design your study and your analyses often, including how you're going to collect the data, how you're going to analyze the data, what statistical inferences you're going to run. You submit that, and then you do all of your work after the fact. And that way you've told people ahead of time. This reduces the kind of ambiguity with, okay, well, did you just make that choice because you found out that your results looked a little bit more interesting because of it? Or did you make that choice for kind of a theoretically, scientifically sound reason? Another option is this idea of multiverse analyses. And so I think with the question Millar asked about this, this convergence approach, this is something that I'm really a fan of. And so Stian and colleagues kind of originally proposed, I think, this term multiverse analyses, but this was taken up by some people, and this explorable multiverse.github.io is a really nice little interactive demonstration of what these look like in practice. And it was proposed by Dragicevic et al. This is a picture from that website. And so what you're seeing here is, a little 2-column data report. All of the blue text in the checkboxes are things that you on the website can click. And as you click them, the results and the figures and the statistics in the report change based on the different choices that have been made. So it's basically, I'm going to run all of the analyses that I can feasibly think of, and I'm going to give you, the reader, the option to choose which ones you think you want to see. And you can play around and see how changing them matters. Kind of the toy examples that they have on that website are toy examples. But this is becoming more and more feasible as journals get on board with this idea of integrating computation and publishing. And so, eLife just last year introduced their first computationally reproducible publication. I am a huge fan of this Distill, which is this machine learning journal that's constantly integrating really cool interactives into their narrative format. And so as journals and publications and publishers kind of get on board with this idea, I think marrying these two approaches of interactivity in publication will become really important to solving these sorts of dilemmas that we face. And then the final option is looking more towards open source tools. So I'm going to highlight here fMRI Prep, because I think that for the types of problems that I've kind of highlighted in this talk, fMRI Prep is a really great solution. So if you've never heard of it, fMRI Prep, you can go online. They also have a Nature Methods paper that was published 1 or 2 years ago. fMRI Prep is a functional MRI pre-processing tool. You give it a data set, it tries to detect different things about your data set and runs what they've considered best practices and gives you an output. And so as more of these sorts of tools become openly available, researchers will be able to kind of flock to them. And instead of rolling your own pre-processing pipeline or analyses, we can converge around a set of tools that everyone as a community can use. And the community can give feedback into what are the best practices that we should be doing, integrate those into these tools, and then kind of as a community, we'll have collectively made these decisions together and have easy implementations of those decisions for researchers to use in their own analyses. So in my mind, the work that I've highlighted today just highlights, a subset of the choices that we can make in a traditional research project. We're making choices every day, even when you don't think you're making choices about an analysis, you are. You probably just haven't thought about it and you're just, kind of going with it. But these three options and things are things that I think can begin to address some of this ambiguity about the research outcomes based on the choices that we make. I'd like to thank my lab and some collaborators, data and funding sources, and just leave you with a few take-home points and some ways to contact me, as well as one final plug for that tool we developed for working with the Allen Human Brain Atlas, Abogen. And that's it. Thanks so much for the time. Thanks so much for dealing with the original room being Zoom bombed and all that.
Yeah, thanks so much. That was really great, and really timely. This is kind of, as journals kind of prioritize these big multi-dimensional data sets in terms of the type of data that people are using. I think it's really exciting to see someone actually worrying about how should we do this integration. So it's a good time for questions. If you have a question, if you don't mind raising your hand, there should be a button at the bottom that allows you to do so. Or you can type in your question here, and you can send it to everyone or directly to me if you want. So while we're waiting, I'll have a couple of questions. If you go back a couple of slides to the three kind of choices that you proposed, the pre-registration choice it takes away one source of ambiguity, but the problem is that you might be converging on the wrong answer, right? Or you might be converging on an answer that the majority of the other methodologies don't let you know. And then on another hand, the multiverse strategy, that also sort of has the same thing, because you can only have so many of these, different analyses that you can stack up in a paper.
Right.
And then how do you actually, speak to those results or discuss them if they actually show very different things, which you show is very plausible. So
right.
what do we do?
So I think what do we do? I think for both of the projects that I kind of discussed, I said that there's no right answer. And I think that that's the case here as well, kind of on a field scientific wide scale. I think that we just need to be thinking about it perhaps a bit more than we are. I think that there's increasingly, as I highlighted at the beginning, research is being done to highlight how these choices are impacting things. But I think that by and large, we still haven't recognized the kind of consequences of that for, all right, take the Allen Human Brain Atlas stuff. If I want to compare the results from some of those published literatures, the published papers using them, I highlighted 4 studies, can I? Are they at all are they convergent? Am I finding similar things? The good thing, I think, maybe, is that in some cases we are, even when people are using different processing pipelines, even when people are doing slightly different analysis or statistical inferences, they're kind of at a hand wavy scale, is some convergence. But it's the devil is in the details. And so I think that we just kind of need to put our heads together and come up with a better framework. I think a lot of that is driven by the kind of incentives of academia: publish new results, get them out there as quickly as possible. And so revisiting some of those things, I think, may have beneficial downstream impacts. But I also think that kind of a focus on computational resources, these sorts of open source tools and things like that make it easier for us to do all of these different analyses, display all these different results would be helpful. Point taken about the multiverse analyses. I think that that's a really elegant solution, but it does grow exponentially based on the number of choices that you have to make in the complexity of your research pipeline. And so I'm honestly not quite sure what we're going to do. I have another couple of years in my degree, and maybe I'll have a better answer for you closer to the end.
That's an excellent answer. Jake Vogel has a question. So first, it's Ross, amazing talk. Thank you for doing this great and useful work. What about the idea of a consensus null? Use several null models and report the results that are consistently significant.
So yeah, I think that that is in the short term something that can definitely be done, and is probably what I would recommend. That idea of convergence in the results that we're getting. The thing is, the things that might be convergent are probably the most robust effects, but oftentimes, especially in neuroscience research, we're working with really subtle differences and subtle effects, depending on the field, depending on what you're looking at, but a lot of them can be quite subtle. People are interested in very subtle interactions. And so this convergence approach may, we may just be showing the most robust effects over and over again. That said, I'm all for it in the interim. I just think that getting everyone on board with that is easier said than done.
Any other questions?
Make a pipeline. So we are this code, all of the code for all of those different null models exists in open source in some form. The reason that I didn't plug one of the tools is just because right now it's just a GitHub repo with all of the code and absolutely no documentation, no way for someone who doesn't know what they're looking at to use it. I fully intend to make all of these kind of analyses available, so it's much easier for any user to say, okay, here's my data, let me run all of these different nulls and get the results. Some of them already have toolboxes. The BERT 2020 method has a toolbox called Brain Smash. Moran spectral randomization is available in BrainSpace, which is also a toolbox. So they already exist, but in terms of the comprehensive consensus approach, absolutely. And if you want to volunteer to help, I'd be more than happy.
I think that's what he's saying. It's not explicitly written there, but I think that's what Jake is saying.
Yeah.
Any other questions? So like I said, this is just, Gabe has a question. What about voxel-wise data?
Yeah, voxel-wise data is definitely one of those tricky things. The only one that can, I don't want to say that, the BERT methods, the parameterized methods can deal with voxel-wise data. So the BERT 2020 paper in bioRxiv right now, just released about a month ago, explicitly shows that their method can accommodate voxel-wise data. They do some analyses using the cerebellum to highlight that. Although the cerebellum can also be modeled as a surface, but they also do some subcortical stuff. And so that is a good point. The spatial permutation models, that is absolutely one of the other weaknesses. The weaknesses, what happens to the medial wall, and what do you do if I want to look at the subcortex? Kind of out of luck there for those. And so this really is a case of the best method to use may be dependent on the question you're asking. If you have voxel-wise data and you're insistent on asking questions with it, then you have to choose a method that can accommodate it.
Great. Nadia asked a question about today's talk being recorded. It will be made available online on the Douglas YouTube channel at some point.
Yes.
I'm working hard on getting all that stuff up. So this week's and last week's actually will both be the first two trials on this. Hopefully soon, apparently. Yeah, so with that, we're already way over time. Not our fault, I'm going to say. Thank you for enduring all the people with nothing better to do and the hackers that were on here a little while ago. And nimbly moving on to this new link. It's going to be an interesting new world, folks. Thank you all for attending. Thanks, Ross, for such a great talk. And if you can unmute yourself and regale Ross and what they deserve, that'd be
Woo! Thank you. Thanks for having me. Stay safe, everybody.
Stay safe. Take care, guys.